So before we can discuss gametogenesis, the formation of eggs and sperm, we have to talk a little bit about chromosomes and what they do. And I do expect that a lot of this lecture is going to be review, but I want to make sure that we're all on the same page before we get going. So we'll start with a look at chromosomes, and specifically chromosome structure. So as I suspect you all have heard before, chromosome structure is defined by two arms and a centromere. The centromere being the part in the middle of the chromosome. I don't care too much that you understand how it's formed or exactly what it does. For the purpose of this course, just know where it is, and then know that the P arm is the short arm, P for petite, and the Q arm is the longer arm. And of course, as I'm sure you know, some chromosomes do not have a P arm or have very short P arms. That's a subject for another class. Something I do care about, though, is that in this picture, both of these structures that you see here are chromosomes. It's just that one of them, the top one, has gone through DNA replication, and so its DNA molecule has been replicated, and now you see two sister chromatids. Sister chromatids are identical copies of each other, okay? Two different DNA molecules, but they are identical copies of each other, whereas homologous chromosomes are two copies of the same chromosome, but one inherited from each parent. And as I know you know, the chromosomes you inherit from each parent are slightly different, and that's certainly what accounts for all the variation we see in humans. So before we go through meiosis and mitosis, we need to talk a little bit about terminology. And something that the book goes over that is important is this idea of ploidy versus N number. So ploidy refers to the number of copies of each chromosome present in a cell, okay? Normal for humans is diploid, so two copies of each chromosome, replicated or unreplicated, two copies of each chromosome. Remembering that a replicated chromosome that has two sister chromatids is still one chromosome, okay? So a diploid is normal for humans, and really for most human cells with very rare exceptions. The diploid number for humans is 46. Human chromosomes come in 23 pairs. So haploid would be one copy of each chromosome, which in humans is 23 chromosomes total. And that's the usual number for mature gametes, eggs, or sperm. Euploid is a term that's U meaning true or true ploidy, which is the usual or appropriate number of chromosomes for a particular individual. So in humans, euploid is diploid. Okay. Now, aneuploid, I'm skipping ahead here a little bit, but aneuploid is too many or too few copies of some or all chromosomes. Okay, so it can be extra missing copies of all chromosomes or of just one or two, but it's not the proper number. Disomy refers to one particular chromosome having two copies. Okay, so if you are disomic, say for chromosome 1, then you have two copies of chromosome 1. As opposed to trisomy, where you have an extra copy of one chromosome. Okay, so trisomy for chromosome 1 means 3 copies for chromosome 1, but you can assume that you have 2 copies of every other chromosome. Triploidy refers to 3 copies of each chromosome. So I have 3 copies of 1, 3 copies of 2, 3 copies of 3, all the way on up through chromosome 22 and the sex chromosomes. Monosomy, our last aneuploidy, is a missing copy of one chromosome, so sort of the opposite of triploidy. So I might have 1 copy of chromosome 1, but 2 copies of all the others. In general, trisomies are more survivable than monosomies, but most aneuploidies are lethal very early in embryonic development. Now, in contrast to ploidy, which is the number of copies of each chromosome, the N number is the number of copies of each DNA molecule in the nucleus. Okay, and so in this picture here, the first chromosome, remembering that both of these are single chromosomes, the first chromosome contains one DNA molecule, so its N is 1, whereas the second has gone through DNA replication and has 2 sister chromatids, so 2 copies of that DNA molecule, making the N 2. Okay. Now, this is sort of a handy cheat sheet. If you're dealing with a diploid cell, you're going to have 43 pairs of chromosomes, or pardon me, 46 chromosomes, 23 pairs of chromosomes, and the N number is on average 2. And then this table sort of goes through the N numbers and how they change with the number of chromosomes. Now, two things that are worth pointing out, okay? The N number is actually going to vary based on the stage, particularly in meiosis. So a diploid cell might have a different N number at a different stage of meiosis. These numbers refer to generally for cells that are not going through cell division. The second is that when you're talking about trisomy and monosomy, where a single chromosome pair has an extra or missing copy and the rest do not, that does not change the N number for the cell. The N number remains the same because it's looking at the cell as a whole and not this one particular abnormal piece. Okay, so for some visuals here, all of these cells shown are euploid and they are all disomic. Okay, so they have the proper ploidy number, which in humans is 2 copies of each chromosome. The first is a diploid cell, okay, 2 copies of each chromosome, and it's 2N because for each DNA molecule we have 2 copies. In this particular cell we have 4 chromosomes but 2 pairs, so the blue represents 1 pair and the green represents a second. In the next cell, still diploid, but this cell has gone through DNA replication, so I still have 4 chromosomes, but each chromosome has 2 sister chromatids, which brings my N number up to 4, because now I've doubled the number of DNA molecules. The next 2 cells are haploid, okay, which for gametes would be euploid, the true ploidy for the cell. So in the first, I have 2 different chromosomes, the blue and the green. They're 1N because I have 1 copy of each DNA molecule, and there are 2 chromosomes present. In the second haploid cell, I'm still haploid and I still have 2 chromosomes, but because they've gone through DNA replication, they're now 2N. Okay, and these pictures are just sort of aimed at giving you an idea of the difference between ploidy and N number and how ploidy can stay the same where N number changes. These are aneuploid cells, but the same idea. So a triploid cell, 3 copies of each chromosome that has not undergone DNA replication will be 6N. A trisomic cell would have an extra copy of 1 chromosome but not of the others, and a monosomic cell would have a missing copy of 1 chromosome but not of all the others. Now what are shown here are groups of karyotypes. Karyotypes are sadly becoming somewhat outdated mode of looking at chromosomes, although not entirely outdated yet. But basically you make a karyotype by taking a cell that's frozen in metaphase, and generally you need to take several different cells from a single cell line so that you can get pictures of all the different chromosomes, but you take pictures of them when the chromosomes are condensed, as they are during during metaphase, and then you can sort of cut them out and pair them up and figure out if you have the right number of each, and on. Problem for another time. But these are karyotypes, and if you've not seen them before and you have questions about them, do let me know. The first is a normal human karyotype. It's disomic, meaning I have 2 copies of each chromosome, which is what I want. The second is a karyotype that you might see for an individual with Down syndrome. So in this case, circled in the red, are the 3 copies of chromosome 21. And it's the 3 copies of chromosome 21 which cause Down syndrome. But because this is trisomy, the rest of the chromosomes have normal copy numbers. Conversely, the third image shows monosomy X, or better known as Turner syndrome, where you're missing 1 X chromosome. In reality, for humans, the default state is female. So if you're missing the second sex chromosome, then you become female, although with some physical differences, and we'll talk about that at a later time. All other human monosomies are generally lethal. Now, just to sort of drive home the point that trisomy and triploidy are 2 very different things, trisomy is occasionally survivable, as is the case with Down syndrome, 1 extra copy of 1 chromosome, whereas triploidy is universally lethal, an extra copy of every chromosome. Now, before we can sort of work our way over to meiosis, which is the type of cell cycle that's relevant for embryology, or more relevant for what we're doing in this class, we need to sort of hit on mitosis so you understand the difference. So again, I understand that this is all review, but it helps to sort of have frame of reference with the cell cycle. First, keep in mind that most cells are not going through the cell cycle. They're often in cell cycle arrest, performing their cell-specific functions. But when a cell's going through mitosis, first it needs to go through S phase, or synthesis, where it replicates its chromosomes. So we go from a chromosome that is not replicated to a chromosome that now contains two sister chromatids, two DNA molecules, two copies of its DNA, but one chromosome. Okay? And as we talked about, then you're going to go from 2N to 4N, but still diploid. Now, when a cell goes through mitosis, as we're about to see, keep in mind that the mother cell and the daughter cells will have the same ploidy and the same N number. Okay, so you're making identical copies of the mother cells. Meiosis is different, and we'll talk about that in a minute. Now again, this is all, I'm sure, review, but this is the level at which I want you to know this for this class. For students who are in the genetic counseling program, I'm sure someone will go over this in much more detail at another time. But we're starting here with a cell, and for purposes of illustration, this cell contains two separate chromosomes, the blue one and the green one, and two copies of each, which is what we would expect. The cell goes through S phase, so now we've replicated the DNA. Then it goes into prophase, so our chromosomes are condensed, okay, which is what's shown there. And then in metaphase, the chromosomes line up along the metaphase plate. Okay, now keep in mind they're not pairing with each other. They're all individually lining up along the plate. During anaphase and then on to telophase, the chromosomes split at the centromere. Okay, so now one of the former sister chromatids is going into one daughter cell and the other's going in the other. But then as the cell divides, we end up with two daughter cells that are both diploid, containing two copies of each chromosome like the mother. And that's mitosis. Now, meiosis is different, and meiosis is special because this is the kind of cell division that goes on when you're making eggs and sperm during gametogenesis. So we start with the same cell with our two copies of our two chromosomes, blue and green. Okay, we still go through synthesis. Thus far, we're the same. But once we get into prophase, we do something a little bit special. So during prophase 1, so meiosis has two stages, meiosis 1 and meiosis 2. During prophase 1, remember that the chromosomes are beginning to pair. So unlike mitosis, the copies of the chromosomes pair together. So one pairs with one and two pairs with two. And when they're pairing, we get crossing over. Essentially, the chromosomes exchange pieces of each other with their homologs. Now, as I'm sure someone will go over in a different class in more detail, the vast majority of the time, this doesn't produce any sort of abnormality. We're exchanging equivalent sections of DNA, but because the two chromosomes who are undergoing exchange are the two copies, one from mother, one from father, that are slightly different in sequence, we're essentially shuffling the DNA sequence to create an entirely unique chromosome structure. And more on that later. For the moment, you just need to know that it happens. Now, metaphase 1 is where things really start to look different from mitosis because now we have paired chromosomes that separate during anaphase and telophase. Okay, and so the daughter cells are now haploid. The mother cell was diploid, but the daughters are haploid. Okay, and this is important because meiosis is a reduction division. Now, when you go into meiosis 2, of course, now remember you've got two cells, okay, and they're both going to go through meiosis 2. And this is, of course, a simplified version, but now, much like in mitosis, in metaphase 2, the chromosomes now line up not paired, because of course they can't, their partner is gone. They now line up along the metaphase plate, and they now split to produce 4 haploid gametes, okay? And of course, we want our gametes to be haploid because when egg and sperm come together, we want them to each bring half of a full set of chromosomes so that the baby has a full set. If gametes were diploid, then the baby would be tetraploid with 4 copies of each chromosome, which is not at all viable in humans. Now, I will point out that what's shown in this picture is more or less what you see for the formation of sperm, for spermatocyte meiosis. When you form sperm from one mother cell, you get 4 sperm cells. But in oocyte meiosis, the formation of eggs, you get 1 haploid gamete and what are called polar bodies, 3 polar bodies. So you really only get 1 gamete out of meiosis when you're making eggs and 4 when you're making sperm. And we'll talk about that more later. Now, helpful cheat sheet for trying to remember the difference. In mitosis, homologs do not pair. The sister chromatids separate during anaphase, and the daughter cells are diploid like the parent. Whereas with meiosis, in meiosis 1, the homologs do pair. In meiosis 2, the sister chromatids separate, so meiosis 2 mirrors mitosis, and meiosis is a reduction division. We want to have half of the chromosomes in the daughter cells, so we want to go from diploid to haploid. And homologs separate before sisters in meiosis. It's alphabetical if that helps at all. Now, of course, we can get errors in meiosis. Those are important too. So the primary error we see is non-disjunction, when the chromosomes fail to separate properly, either the paired homologs or the sister chromatids. Okay, and this is a problem that becomes more common in eggs as women age, and we'll talk about why, but it has to do with the idea that eggs are present in the woman from the time she is a fetus. They arrest in meiosis and then begin meiosis when they ovulate. So they've spent all this time in stasis, and the mechanism that's separating the chromosomes has essentially gotten old. Now, what this diagram is showing you here are 2 different types of non-disjunction error, because of course it can occur in meiosis 1 or in meiosis 2. Now, the important thing here is that if it occurs in meiosis 1, okay, then what we've got are the maternal and paternal copies of the chromosome traveling together into meiosis 2, okay, and then failing to separate. Whereas in meiosis 2, a meiosis 2 non-disjunction error, the maternal and paternal chromosomes do separate, but the sister chromatids fail to separate at meiosis 2. And you can actually tell the difference in some cases with genetic testing. Okay, but either way, the end result for the baby is the same. In this case, it would produce Down syndrome. And if you have some time, have a look at this table and watch how the risk increases for women as they age. And what I want you to notice is that, yes, it does increase, but at no age does it jump. Okay, it's not as if a woman turns 35, the commonly accepted cutoff for being of advanced maternal age, higher risk for this kind of error. It's not as if you turn 35 and the risk skyrockets. It goes up a little bit every year. Now, the primary types of aneuploidies that we end up talking about in a clinic are the ones that are survivable. And so, of course, we do talk about Down syndrome, and most of you are probably familiar with Down syndrome on some level. The scientific name is trisomy 21, an extra copy of chromosome 21. It is almost always sporadic, although it can run in families under different circumstances. And I'm sure you're familiar with many of the features of it. The one thing I do want to point out is that when you're in a class like this, or in any sort of class where you go over a condition like Down syndrome, you often get these kinds of pictures that highlight the different features that go along with the syndrome. But what parents really want you to know, and what families want you to know, is that behind those pictures are children leading actual lives with actual families, and it's important not to lose sight of that. And in fact, if you have the time, it's worth going to the blog that this particular picture came from. The link is in the upper left-hand corner. That will give you a great perspective, not on Down syndrome as a medical condition, but on living life in a family where somebody has Down syndrome. The other one that I wanted to highlight is trisomy 18. It does have a common name of Edwards syndrome, although it's almost never used. This one is almost always lethal, but not always before birth. Okay, sometimes these babies are born alive, although if they are, they usually don't live very long. In the picture on the right here, these are twins where one baby has it and one does not, and you can see that there is a significant difference between the 2 of them. Now, occasionally a baby will be born with a partial or mosaic form of trisomy 18 or another aneuploidy, And those forms may be milder and survivable in part because the extra piece, or in some cases missing piece, is either only a portion of the chromosome or not every cell has it. Which brings us to errors of mitosis. So these are not the kinds of errors that generally produce anomalies in embryology, although occasionally they can, okay? But these mitotic errors would be errors that occurred after conception, okay? So sometimes it's shortly after conception, sometimes it's well after conception. But what we've done here is create two different cell lines. So if we have an error in mitosis shortly after conception, okay, the normal mitosis, if it went through normally, we would expect to have an embryo where every cell has the same number of chromosomes. But if you get a non-disjunction event early on in mitosis, in the development of an embryo, you can get an aneuploid embryo. But what's really interesting is if you have this event occur shortly after conception, when you already have a population of some cells that have normal numbers of chromosomes, and now you introduce a population of cells that have an abnormal number of chromosomes as a result of one non-disjunction event, you now have a mosaic embryo where some cells have the extra chromosome, hypothetically, and some do not. And that tends to produce a milder condition than the full aneuploidy, because some cells are unaffected. But it's very difficult to predict severity, even if you detect something like this prenatally. And so these children show a great deal of variation in their symptoms. But on occasion, you will see an aneuploidy that you don't normally see in live-born children, like, for example, trisomy 12, in a child that is mosaic.